Okay, ladies and gentlemen, so this is going to be your notes on biochemistry. I am breaking this down into 3 videos. This first one will be just some basic overview of, we'll go with functional groups and how subunits are combined together and things like that. And then the subsequent 2 videos will be about the specific macromolecules we're talking about. Okay, so biochemistry is simply the study of the chemistry of life. That sort of makes sense, right? We've got bio and chemistry in there. It describes structure, mechanism, interaction between molecules and chemical processes shared by organisms in molecular terms. Okay, so we're looking at really, really low. In biology, we always start with the smallest subunits and we work our way up. So that's why we start the year with biochemistry. There are fewer than 25 elements that are actually essential to living organisms. You can see here in this cute little graph or a depiction of our periodic table that our bulk elements are in sort of that tannish color, and then our trace elements are in the yellowish. One of the ones that I always like to point out, for example, is that iron is, I have a laser pointer on here, sorry, really just easily distracted. That iron here is one of our trace elements that we need. We're more familiar with that probably than most of the other trace elements that we are accustomed to seeing here. Okay, hydrogen, oxygen, nitrogen, and carbon make up greater than 90% of the mass of most cells. This leads to one of our fundamental pieces of evidence to sharing a common ancestor, is that all organisms share, that there's a universality to chemical intermediates. Okay, how a maple tree digests sugar and how humans digest sugar is not all that different on a fundamental chemical level, which is sort of that indication that these really important processes have not changed a great deal in our evolutionary time frame. Okay, biomolecules are carbons, or are compounds of carbon. Okay, you watched, hopefully, the Carbon is a Triumph video. Hank Green did an excellent job of explaining here, right? It's versatile. We can have straight or branched chains. You can have it form rings. It's capable of forming 4 single bonds. Conversely, it can also form multiple bonds. You can have it be single bonded on one side and triple bonded on the other. Like, it's just this magnificent molecule. It is, as Hank Green mentions in the video, we have a hard time comprehending life forms that are not carbon-based because its size and its versatility just make it so useful. It is the black Sharpie marker of the element world. Okay, so then we have functional groups. These are particular arrangements of atoms that are often attached to a carbon backbone, and they help determine the characteristics of a molecule as well as its chemical reactivity. Okay, how does it bond to other things? How quickly does it bond to other things? That sort of stuff. There are a lot of functional groups. You have to know them. I'm just telling you straight up that this is something that you are going to need to know. I will be asking you it on your test. I will be asking you it on the midterm. I will be asking it of you on the final exam. You must know your functional groups. Okay, so hydrogen. Almost all organic molecules have hydrogen, and all that is an H hanging off at the end here. It is involved in hydrolysis and dehydration synthesis, which we are going to discuss towards the end of this particular video. Hydroxyl, which is OH, this is the other half that's involved in dehydration synthesis and hydrolysis. You find hydroxyl groups in carbohydrates, nucleic acids, alcohols, some overall, just some acids, and steroids also have it. I had a really crazy dream in undergrad one time that there was an alcohol group, which is an OH, in mass spec, you refer to it as an alcohol group, attached to my alarm clock, so I didn't wake up in time for my organic chemistry test. It was all sorts of crazy. A carboxyl group is COOH. How this traditionally looks, by the way, is I have my carbon, and then I double bond it to an oxygen up here, and then I have an OH off to the side here. Sorry, I'm not particularly adept at writing on my laptop, but So that is traditionally what a carboxyl group actually looks like. And carboxyl groups are involved in amino acids and fatty acids. It is acidic. They are involved in peptide bonds. We'll talk in more detail later, but peptide bonds are what are found in proteins. The amino group, NH2, is the other half of a peptide bond, by the way. It is basic, non-acidic, and also involved in amino acids. It's also involved in nucleic acids, okay? Very important in nucleic acids, the amino group. Phosphate, which is a PO4. This is the nucleic acids. When we talk about DNA, we'll talk about our sugar phosphate backbone, and then you have the nucleotides are the rungs in the ladder, but the sugar combined with the phosphate is sort of what makes up the outside of that double helix. They're also involved in phospholipids, which we'll talk about in a little bit, which make up our cell membranes. It's acidic, it links nucleotides to nucleic acids, and it's an energy carrier in ATP. Okay, so you add a phosphate, when you have 3 phosphate groups, you have ATP. When you knock one off, you release a whole bunch of energy and you have ADP, which we'll talk about in much more detail when we talk cellular respiration. Methyl group, which is CH3, this is common in lipids. It makes molecules hydrophobic. Hydrophobic means water-fearing. Okay, so isomers. Isomers have the same number and kind of atoms, but they have a different arrangement of those atoms. So for example, glucose and fructose are both C6H12O6. Sorry, PowerPoint did not let me type that nicely. That's whole wonky wrong. Remember, you have capital letters and you have, although those are capital letters, but you have subscripts. Anyway, if you look here, you see that glucose is a pentose. Or I'm sorry, is a hexose sugar structure. Okay, it's 6-sided, as opposed to fructose is a pentose sugar. Hint, hint, you're going to have to know the difference between those two. This is your friendly reminder. Okay. My 4 major types of organic molecules. I have polysaccharides, which are simply polymers of simple sugars. These are carbohydrates. Okay, that's just another way to say it. 2 major functions. They are energy yielding fuel stores and extracellular structural elements, so they help sort of hold things in place. Lipids, which are an oily hydrocarbon derivatives. These are structural components of membranes. They're an energy-rich fuel store. They're found in pigments. That's why a lot of pigments stick to clothes really nicely, is because they are oil-based, and they are also involved in intercellular signals. Proteins are large polymers of amino acids. They act as enzymes, which are catalysts. Remember that a catalyst is something that is used to speed up a chemical reaction, but not consumed in the chemical reaction. They're structural elements, and they're signal receptors and transporters. And then nucleic acids are polymers of nucleotides, and they store and transmit genetic information. So in order to form these, okay, we start with monomers. Mono means one, okay? Poly means many. When I'm Empress of the Universe, everybody will be required to take Latin. So a monomer is a single subunit, a polymer is many monomers put together. So this is just a nice little chart of what our different subunits can make. I'm not going to go over it in excruciating detail. You can always come back to it, okay? So in order to make polymers, I need dehydration synthesis. So think about it this way. Dehydration means to take out water, right? If you're dehydrated, you don't have enough water, and synthesis means to make. So this literally means to make by removing water. So it's a chemical reaction that links monomers to form larger molecules. I remove the hydrogen from one subunit and a hydroxyl from the other subunit. Remember that hydroxyl is an OH. A covalent bond is formed, and the free hydrogen and hydroxyl combine together in order to form water. I recall this link works. I'll make sure that you actually have a live link in the Teams app. So when I look here, I have my hydroxyl over here and my hydrogen over here. Those go out to make my water. And now this carbon, I'm going to put a star next to it. Okay, this carbon now forms this nice little bond with the nitrogen that is over here. This is an example of dehydration synthesis in order to make a polypeptide. This is our carboxyl group, and this is our amino group. Now, hydrolysis is the opposite. Hydro means water, lysis means to break, so we're literally now breaking with water. It's the reverse of dehydration synthesis. I split molecules by adding water and it breaks the covalent bond. So now I have my monomer here, right? I've got this bond in between it. I add water to it. Now my hydrogen is over here. My hydroxyl is over here. The only reason I know that, folks, by the way, off the top of my head, is I can see here there's a hydrogen over here and a hydroxyl over here. So that indicates to me what order it should be. And that's not something you need to necessarily have memorized. And then I look and I break this bond also. And now I have my 3 subunits. So when, you know, we're trying to digest food and get this to a fundamental level, that's what we're doing. And that is going to be that for the first day of notes. Come back for day 2.